Hi everyone, I'm Iris and today I'm going to present on the role of beta-adrenoceptors in distal ureteral relaxation. And this is our work from the Center for Urology at Bond University in Gold Coast, Australia. So urolothiasis is more commonly known as kidney stones, and these stones are formed in the kidneys but have been shown to always lodge in the ureters, which are the tubes that connect the kidneys to the bladder. So what happens is the stones actually gets constricted in the ureters and therefore causes an excruciating pain to the patients. So the treatment options for urolothiasis, there are many and it's highly dependent on the stone size. So one of the most, I guess, when it's a very large stone, what would happen is we just use surgery. But there are other options as well, like shockwave lithotripsy and ureteroscopy. But these treatments are considered invasive treatments, and therefore in the recent years pharmacological treatment for kidney stones have been more I guess a bit more popular because it's less invasive. So the most commonly used medical expulsive therapy where pharmacological treatments are used to relax the ureter is tamsulosin and nifedipine. Well, nifedipine is not highly used anymore, but tamsulosin, which is an alpha-1 antagonist, causes relaxation of the ureter, dilating the ureter and allowing the passage of the stones into the bladder much easier. So our research aim in this study is really to look at other targets for pharmacological treatments of stones, and therefore looking at the pharmacology of the ureter. So in this particular study, we are looking at the role, or whether there is a role for beta-adrenoceptors in distal ureteral contractility, and seeing if the beta-adrenoceptors can't relax the ureter, because this has obviously been seen in the bladder and also the urethra, showing that beta-adrenoceptors do play a role. So in this study, we used the usual organ bath method, where we suspended isolated distal porcine ureter into the organ baths into Krebs solution and measured the tension of the contraction. So what we did first for this study is subjected the tissue strips to 100 micromolar of 5-HT, which allowed the tissues to then develop a sustained contraction, and I'll show you in the next slide what the contractions look like. But what we did then was, in paired tissues, we examined these contractions in the absence or presence of increasing concentrations of isoprenaline, which is a general beta-adrenoceptor agonist, mirabegron, which is a beta-3-adrenoceptor agonist, and then CGP compound, which is another beta-3-adrenoceptor agonist, and also beta-2-adrenoceptor agonist, salbutamol. So this here shows what the contractions look like after it's subjected to 5-HT. So as I said, we allowed the contractions to stabilize before we added increasing concentrations of any of the beta agonists. So the parameters that we measured based on these contractions were three things. So the first one being the frequency of these basic contractions, the second one being the maximum amplitude of these contractions in that particular period of time. And then lastly, we also looked at the area under the curve to basically get a general idea of the general contractility involving both frequency and amplitude. So these graphs here now show the response to isoprenaline. So the dotted lines is actually just a vehicle control, or I guess a time control, you could say. So you can see here that isoprenaline with increasing concentrations did cause that relaxatory response. So it reduced frequency, reduced amplitude, and also reduced the area under the curve of the contractions. So the next thing we wanted to look at was to see which beta-adrenoceptor subtype was the one that mediates this isoprenaline-induced effect. So therefore, we looked at beta-3-adrenoceptors first, and this was the expected one because it has been shown obviously to be functionally present in the bladder, which is in close proximity to the ureters. So we looked at mirabegron in this first one, which is a beta-3 selective antagonist, and you can see here that mirabegron actually didn't have those effects. So it didn't reduce the frequency or the area under the curve, and you can see that even when it reduced the amplitude, it was in very high concentrations, which does not really, it's not really selective for beta-3 at that point. So that was beta-3 adrenoceptors. In addition to mirabegron, we also actually did CGP compound, which is also another beta-3 adrenoceptor agonist, which I haven't shown the results here, but it showed pretty much the same thing, where it didn't have any relaxatory effect. So then we looked at beta-2 adrenoceptors, and we used beta-2 adrenoceptor agonist salbutamol, and did pretty much the same thing to check if it would reduce or have any relaxation. And very similarly, you can see here that salbutamol also was only able to reduce the amplitude and the area under the curve of these contractions at very high concentrations, which again just meant that it wasn't where it was very selective for beta-2 adrenoceptors. So therefore, we were pretty certain that it wasn't via beta-2 or beta-3 adrenoceptors. So in conclusion, what we found was that beta-adrenoceptors do have a functional role, because isoprenaline was able to reduce significantly the frequency and also the amplitude of phasic contractions of the ureter, but then the inability of mirabegron and salbutamol to reproduce this relaxation just showed that it wasn't beta-3 or beta-2 adrenoceptors. Thank you very much, and please feel free to ask me any questions.